Now, okay, now the following lesson will focus on mutations and the changes that they entail. Now, this lesson will introduce some of the causes of mutation before describing what they entail. This lesson will also cite some different types of mutations, point, silent point mutations, silent mutations, and frameshift mutations, and explain their possible effects. And finally, this lesson will conclude with some of the genetic disorders, along with the groups in which these disorders are most prevalent. Now, first off, before going further, I would like to ask a question when it comes to mutations. What normally comes to mind when you first think of mutations?
Genetic surprises or abnormalities?
Mm-hmm. Yes, genetic surprises or abnormalities do come to mind. And generally when it comes to the American public, when it comes to what the American public thinks of mutations, given that there's often a lot of popular stereotypes and misconceptions about mutations, or some popular stereotypes that come about when people think of mutations, or when you mention mutation.
Either disease or special abilities, like in a comic book movie.
Mm-hmm. Yes, whenever it comes to mutations, I used to think of people with special abilities. Look at all of the superhero movies that are coming about. In fact, that's a staple of the superhero movie and comic book industry. I also would like to think of mutations as what you would run across in a post-apocalypse environment, especially a post-atomic war environment where mutations create all sorts of powerful monsters, and those humans who are fortunate enough to survive a nuclear war find themselves knocked down the food chain a little bit because they're dodging cockroaches the size of SUVs and things like that. Now, the reality of mutations, though, and while they do create serious changes, the causes behind them, ironically, are somewhat more mundane. Now, when it comes to mutations, the surprising thing about a mutation is the basic cause behind it is relatively simple. All mutations involve are a change in the nucleotide basis of a DNA sequence. Basically, all you have nucleotide sequences of the DNA, and you just have a change in one or more nucleotides, and that's what a mutation involves. Now, lots of things cause mutations. Surprisingly, some of these things are very mundane. Of course, viruses can cause them, along with chemicals and food. In addition to viruses, chemicals, and foods, there of course is the exposure to ultraviolet radiation. What's also surprising, though, is that mutation can result from more mundane things like exposure to excess cigarette smoke, or things like drug or alcohol intake, or exposure to industrial or automobile emissions. My understanding is that this would be especially common when it comes to babies in the womb who might be exposed to drugs, food, radiation, or if the mother is unfortunate enough to inhale industrial pollution or automobile fumes or things like that. So there are lots of things that can cause mutations, and we're learning more about them as you do more research. Now, mutations involve changes in DNA nucleotides, and the reason they produce all manner of effects are that when the DNA nucleotide changes, what happens is you get a change in the messenger RNA sequences, specifically the change in the messenger RNA nucleotide. Now, what happens is that a mutation is a two-step process in terms of how it produces effects. First off, the mutation is going to involve a change in the DNA. DNA will then transcribe a different messenger RNA. And when the different messenger RNAs are transcribed, what then happens is that altered messenger RNA is going to be translated, and depending on the change, that messenger RNA can produce a different type of amino acid. And that's what the mutation entails in terms of its physical effect. It's when a new type of amino acid is created. Now, what's interesting is that in some cases you may have a new type of amino acid created, or you might have no amino acids at all, because the mRNA could be a stop codon where the chain of amino acids that would result from a non-mutation, well, suddenly that chain is terminated because alteration produces a stop codon. Now, you might have completely altered proteins or no protein at all, depending on the messenger RNA sequence. Or you may also have a messenger RNA sequence that changed, but the same type of amino acid results. And if the same type of amino acid results, despite a different messenger RNA combination, that mutation is not observable. It still is mutation because mutation is technically a change in DNA, but in this case, mutation might not be observable at all. Now, when it comes to mutations, you'll first have to focus on replication, or the creation of daughter DNA from a DNA strand. Now, mutations are going to start out as alterations of nucleotide-based sequences on the DNA. Now, during DNA replication, what happens is that typically adenine will bind to thymine, and guanine is going to bind to cytosine. And this diagram right here shows how adenine will bind thymine and guanine will bind cytosine. Now, normally in replication, you'll have guanine binding to cytosine, but in the case of mutation, you've had the cytosine replaced by thymine. Thymine is not supposed to bind to guanine, but thanks to a mutation, you have that change right here, where thymine is now bound to guanine. Now, alterations will lead to an altered nucleotide base sequence. In this case, thymine has replaced cytosine to bind guanine. Now, these altered base sequences in the DNA are going to start to impact the organism when the DNA transcribes a different messenger RNA. And what happens is when you have this different messenger RNA, that different messenger RNA will be translated to create a different amino acid. And it's when this different amino acid appears that you're going to have your observable and noticeable effect of mutation. Now, what happens is, when it comes to describing the transcription translation process more specifically, when DNA transcribes messenger RNA, the sequences normally transcribed will involve the adenine on DNA binding to uracil in place of thymine on the RNA, while the guanine binds to the cytosine. But if the DNA nucleotide is altered, you'll have a messenger RNA with a different sequence. Now, that being said, are there any questions so far as the way that DNA can be altered in the process of replication, or what this alteration has when it comes to messenger RNA? Are there any questions concerning this thing?
No.
Okay, if there are no questions, then what happens is that once messenger RNA is transcribed, it will then travel to the ribosome and it will bind with transfer RNA, and once these two different RNAs couple, you're going to have amino acids brought in, and in the ribosome, these amino acids are going to start to form a chain, and all a protein really is, it's just a chain of amino acids. That's what proteins are. Now, what's surprising about a protein and an amino acid is how simple they really are when you think about it, because at the most basic level, each amino acid has just a unique 3-nucleotide sequence which the messenger RNA codes for. And this is a chart right here of the different nucleotide sequences for the different amino acids. There are 20 different amino acids, and each of these amino acids have at least one 3-nucleotide base sequence in the messenger RNA. Now, what's interesting is that several amino acids have multiple messenger RNA combinations, and that's what makes mutations somewhat interesting, because thanks to these different messenger RNA combinations, in some cases the mutation could have no effect because the same amino acid is still coded for despite the different sequence. So it's important to keep in mind that an amino acid is just composed of 3 nucleotide sequences found on the messenger RNA. And an alteration of messenger RNA could result in a different type of amino acid or the same one. Now, the 3 nucleotide sequence on the messenger RNA is referred to as the codon, and when it comes to complementary sequence on the transfer RNA, it's called the anticodon. Basically what happens when an amino acid is synthesized is the codon and anticodon will interact to bind together, and then the transfer RNA will bring the amino acid into play, and then another transfer RNA and messenger RNA sequence will appear to create a whole chain of amino acids. Now, exploring how an altered amino acid sequence, how an altered messenger RNA leads to a different amino acid result, consider this sequence. And this is a DNA sequence right here, which codes for a corresponding RNA sequence. Now, what can happen is that sometimes chains can appear to be rather mundane. What can happen in this case is that the second codon on the DNA sequence, ACC, is replaced by ATC, so you have a thymine replacing a cytosine in the second 3-base sequence on the DNA. Now, a very minor change on the surface, but what happens is that the resulting messenger RNA is going to change to UAG on the second codon. Now what happens is that this is very serious change because it's a stop codon, and so what's interesting is that when you have the stop codon, originally you had about 4 other amino acids that were going to appear, but when you have a stop codon, it's like those 4 other amino acids are terminated and they never existed. So essentially, the original protein comprised of 4 amino acids suddenly no longer exists because the translation of those 4 amino acids is completely stopped, and that just results from simply one nucleotide base on the DNA being changed. Now, this is a classic example of how a single change in the nucleotide can create all manner of complications and things like that. Now, that being said, are there any questions as to how change in just one nucleotide of the DNA can completely wreak havoc with respect to amino acids being altered or not even existing? Okay, now when it comes to mutations, there are different names for mutations. The mutation we saw earlier on is a point mutation. A point mutation involves one nucleotide replacing another, and what's interesting about a point mutation is it may or may not lead to an altered amino acid. In this case, the point mutation resulted in a stop codon, leading to a termination of amino acid production. Now also, what can happen is that a point mutation can lead to a different type of amino acid. In this case, the 4th codon is going to be altered on the messenger RNA, and when this 4th codon is altered, that new amino acid is going to be tryptophan instead of cysteine in the original sequence. So you have the same original chain of amino acids except for one which is altered. Now, also though, there are cases where an altered messenger RNA sequence may not lead to any change in the amino acid. What can happen is that you can have a change in the nucleotide sequence of the messenger RNA thanks to a DNA change, but what happens is that the amino acid remains unchanged, because if you remember from that chart, some amino acids have as many as 4 different combinations. So one change or the other in the nucleotide sequence, if an amino acid has 4 combinations, chances are it's still going to be there. Now, in this case, while the mutation still is there because of a change in the DNA, you have the same amino acid, and these mutations are referred to as silent mutations because no changes are observed. And then there's another more complicated mutation, which is the frame shift mutation. Now with a frame shift mutation, nucleotide base is slipped or deleted into the genetic code, and what happens when you have a single base either deleted or inserted is that the entire sequence of codons is going to shift to the right or to the left, and so what happens is you get an entirely different string of amino acids. It's sort of like the case of reading the sentence, watch Becca chase the cat, but you insert another letter in the midst of that sentence, and then the sentence is completely screwed up. That's kind of like what frame shift mutation does when it comes to alterations of amino acids. And a case of a frame shift mutation would involve this nucleotide sequence where you have C that is going to be inserted after the third codon before AAC, and then what you have is you're going to have it read after UGC. It's going to be CAA rather than AAC, and then these two CG are going to be thrown together, and then CUG is going to be thrown together, and the C is going to be booted out, and so you have 4 different amino acids resulting from this frame shift mutation. And of course, when you have 4 different amino acids, that's going to result in all sorts of big screw-ups for the protein. And in this case, you have 4 different amino acids resulting. And of course, what happens is you get a completely different protein. And this example right here is going to illustrate how a frame shift mutation will completely alter the amino acid sequence and the resulting protein. Now, that being said, are there any questions about what point or frame shift mutations entail? Any questions about those?
No.
Okay. Well, if there are no questions, then we're going to go into a few of the better known mutations. And one well-known mutation is sickle cell anemia. In this case, you have the hemoglobin S gene that's mutated, and as a result, the red blood cells will have a sickle shape instead of a round shape. And what happens is, in this case, it's a result of a point mutation where you have exactly the same amino acid chain in people with sickle cell as opposed to people who are not affected, but you have one amino acid that's changed. In this case, you have valine being substituted in place of glutamine for the amino acid. And what happens when you have valine substituted for glutamine is that the red blood cell will shift from a round to a sickle shape. And what does that do? Well, as you probably would guess after looking how blood cells flow, it will impede the flow of red blood cells carrying oxygen to the tissues, and so people with sickle cell will suffer from pain, fatigue, and dizziness. They'll also suffer from low oxygen, and they'll tire out pretty quickly. But in some cases, what we would regard as a curse can also be regarded as an advantage, because in regions that are high in malaria, this trait can convey resistance to malaria, and somebody with sickle cell can be at something of an advantage in a malaria region, especially if only one of their parents has a sickle cell trait. Because if somebody has only one parent that has sickle cell, then some of their blood cells are also going to be round, so they'll circulate almost like normal, and it's going to be a case of round red blood cells intermixed with sickle-shaped red blood cells. And that will give people in malaria regions something of a health advantage. And this disorder is most often occurs among Africans and African Americans. Now, another common mutation is cystic fibrosis. In this case, the CFTR gene regulating the passage of water and a salt into and out of the cells is disrupted. And what happens is when this passage of water and salt out of the cells is disrupted, they're going to have sticky mucus and sweat, which is going to impede the air passageways. And people who suffer from cystic fibrosis will have lung infections. They'll also have difficulty breathing, constant fatigue and coughs, along with inability to gain weight. And people with cystic fibrosis will sadly rarely live beyond their 30s, but this condition can be treated. People will be able with cystic fibrosis can prolong their lives, but they often have to carry on portable oxygen tanks, and this can also be treated through lung transplants. And when it comes to occurrence, this disease is most prevalent among people of Northern European lineage. And another common genetic disease is phenylketonuria. This is a disorder where the body cannot synthesize an enzyme that breaks down phenylalanine. And what happens is, if this disorder is untreated, it will result in brain damage, learning disabilities, and slow growth because of the buildup of phenylalanine, which the body cannot break down. Now, this disorder is most common among the people of Northern European lineage. But an advantage of when it comes to phenylketonuria is, as a disease, it can be identified in infancy and it can be treated. Now, a more serious genetic disorder is Tay-Sachs disease. This involves a mutation of the gene so it cannot produce beta hexaminidase enzyme, and beta hexaminidase, without that enzyme, this is going to result in destruction of nerve cells of the brain and spinal cord. And as nerve cells of the brain and spinal cord are destroyed, there's going to be muscle weakness, hearing loss, and difficulty swallowing. This condition will start in infancy, and it will worsen in infants who are born with Tay-Sachs disease will almost always die by the age of 4. And this disorder is most prevalent among those of Ashkenazi Jewish descent. One of my friends from a church I used to go to, she was being screened for Tay-Sachs disease because she was of Ashkenazi Jewish lineage, and she wanted to make sure that it would be safe to have children, so that she was not carrying this possible disorder. And finally, there's Huntington's disease. Huntington's disease is a genetic disorder we have started to study a little more, and this is a disease that appears in the middle stage and later in life, and unfortunately there is no known treatment for this disease at this point. Symptoms of Huntington's will include memory loss, involuntary movement, loss of muscle control, anxiety, or depression, because this disease is going to involve a breakdown of the nerve and the brain cells. Now, that being said, are there any questions about mutations or genetic disorders?
No, I don't think so. Thank you.
Okay. Well, I'd like to thank you so much for all of your support, and please feel free to ask. I have only limited knowledge of genetic disorders and mutations, but this is very much an interesting subject of research and future endeavor.
Thank you, Mr. Lockett.